Hey, somebody play the damn video. Hey, thanks, guys. I told you to play the damn video. Nobody's listening to me, man. I'm not appreciating it.
Hit the lights.
Yeah. Whatever. Yo, 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 what it is, this is Nerd Night! Good evening, my name is John Matthews. I will be your MC, host, and probably not your damn friend. Welcome to Nerd Night. We are always happy to have all of you here, some returning speakers. There's a few of y'all in the crowd, some regulars. Thank you all for coming. And the rest of you, where the hell have you been? First timers at Nerd Night, raise your hands. First timers, damn, that's a lot of y'all. Hey, first of all. Second of all, 3 speakers walk in, 3 speakers walk out. We're not savages. There will be science, he talks. Yeah, I used that joke last time, I'm really sorry. Or was that 2 times ago? Or was it every time? Maximum 3 times. For those of you joining at home, what's up, hey. We are streaming from Tokyo. It is Friday someday. My smartwatch doesn't have the date on the front screen, so who cares? December 7th, I think it is today. Anyway, so we are streaming live. If you have friends, we have a stream link on our Facebook page. Our Facebook is facebook.com/nerdnite, N-I-T-E, Tokyo. We're on Twitter too, but we're not consistent with our naming, so y'all can figure that out for yourselves. What else was I supposed to talk about? Shit, I got caught in that trap. Boss Amanda yelling at me as per usual. Firstly, next time we come back here will be December 7th, not today. December 7th is the next time. We're talking about yaoi, manga, which should get most of you back in the room. Japanese science films, because you've had enough of Western stuff, naturally. And I'm going to say distributed apps, to be a little more correct, perhaps. Lots of cool stuff to talk about, cool stuff as per usual. If you enjoyed tonight, please come back and see us next month, December 7th. The year of our Lord 2018. We'll be right here at Nagatacho Grid. Again, please follow us on Facebook and on Twitter, or just one, or both, or neither. It's really up to you. And I'm going to look at my thing for the next thing. Yes, speaking of which, speaking of cool speakers, if you can do 20 minutes on something you enjoy, that you know a lot about, doesn't have to be science, doesn't have to be whatever. We had one of my favorite talks so far was Craig's, which is Andrew's not looking, but he'll be able to talk about it, which he's not going to because he doesn't have a microphone. We had a guy talk about pentameter, not pentameter, but what's the term, Andrew? Theater. No, it wasn't that. It was not rhythmic, something else. Anyway, it was about something that wasn't science. What? Something like that. Anyway, language arts, science, nerd stuff, video gaming. I'm planning on doing a VR demo with dear friend and developer and human, Don, and longtime supporter. Here in the near future, we do really cool stuff here. Please come back and see us again soon. Again, follow us on this stuff and we'll tell you when we're coming back. I believe that leads me to the idea of having cool merch. We've been doing this for 2.5 years-ish, more or less. Actually, almost exactly 2.5 years, actually. And we've got some cool merch. They're beakers. I have one at home. I paid for it, I promise. We do have really awesome beakers. They are 1,500 yen. At the bar, yeah. Step forward. Step closer, 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 closer. I don't own the space. Come stand up closer. In the light. 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 Yes, this is Ian, friends. Clap for Ian. Ian. Mm-hmm. Ian is your bartender. He comes with a special tool of glasses, I guess. But the beakers are 1,500 yen, so please go see him to buy some. We have how many left? Like a dozen max? Yeah, we don't have a whole lot left from the first run, so go get them now, else you'll have to wait for Japanese efficiency to kick in. Right, that was pretty mean. I've been here 11 years. It's okay, I swear. So, let's go ahead and talk about tonight's speakers. Get them in here so you can stop listening to my flabby-gabby mouth. First, we'll be having Daniel Jeans. Jeans as in Levi's jeans, he tells me, so I'm going to pronounce it correctly this evening. Thomas Chater will come after that. Lights, camera, calcium, much more interesting to me. I'm just kidding. I have no idea what he's talking about. And then Ade Suriajaya, who we call Cha-Cha. We have an absolutely fascinating discussion about Zeta functions. And if you know anything about them, you'll probably really enjoy this. And if you don't, you'll probably really enjoy this. She's right there. I'm making fun of her. Anyway, are we ready to go? Is Captain Beardy ready to go? Daniel, are you ready? Where are you? Where are you? Should be right here in front of me. Yeah, come step up here. Step up here, Daniel. Our first speaker tonight, Daniel Jeans, hails from Cambridge, the UK, naturally. I don't know of any others. There might be some like American town sort of pithily named after it. No imagination whatsoever. He, however, is a physicist. He's been working in Switzerland, Italy, the US, France, and Japan, this lovely country. He's currently an associate professor at KEK, I almost said Keck, and would have been very, very dumb saying that, the Japanese High Energy Accelerator Research Organization in Tsukuba. Surprise, surprise, his interests in research are high energy physics, Higgs particles, and particle detectors. And now that he has his clicker, I'm going to hand him over to you. Everybody, please applaud for Daniel Jeans.
But maybe there's another dimension in space in which it does spin. There are also theories which predict this sort of effects. So there are many questions we have about the Higgs, so that's why we think it is worth looking at it. So that's where the International Linear Collider, the ILC, comes in. This ILC is basically a factory which will produce Higgs particles in abundance. So over a period of maybe 10 years or something, or 20 years, we'll produce something like 1 million Higgs particles and really probe them and measure them as well as we can to try and understand some of these questions, the answers to some of these questions we have. For example, how are these Higgs particles produced? How long are they going to live? How long they did very quickly, but we don't know exactly how quickly they will decay. How do they die once they reach the end of their lives? Higgs particles really, in our theory, a Higgs particle is just an elementary particle, but maybe this Higgs particle is made out of other types, new particles we haven't even thought about. In a similar way to the proton is made out of quarks, for example. Is there only one type of Higgs particle, or are there several different types with different properties? We don't know. We want to find out. So how does the ILC work? So the ILC starts off with an electron and an anti-electron, to which we give as much energy as we can, and then we bang them together in the center. So we bang these together, and thanks to this nice little equation here we talked about at the beginning, we make some pure energy. And then if we are lucky, this energy can occasionally make a Higgs particle. It makes a Higgs particle together with another type of particle, but what's important for us is that occasionally we can make a Higgs particle in this sort of collision. It's not so trivial to do. So in the ILC, we'll take 100 million electrons, 100 million anti-electrons, and collide them every second. So that's a lot of collisions, I think you can imagine. But we only get one of these Higgs particles every hour. So it's a rather inefficient factory, but that's the best way we have to make it in our labs. So what does it look like? It looks like this, so it's around 20 meters long. And it has a gun to produce electrons over here. They get whizzed around and then taken to the end of this long tunnel and then accelerated in this high energy, where they then later will meet the positrons, the anti-electrons, coming the other way. So this machine has been studied for a long time, maybe 20 or 30 years now. People have been interested in building such a machine to study the Higgs and other particles as well. And now, maybe around 5 years ago, we completed a major design of such a machine, and we understand well how to make it. And now that this Higgs particle, we know it really exists, this gives us a very strong impetus to actually build this machine now so we can study this Higgs particle. So next, I would like to show the little movie, please, Mr. Technical Coordinator. So this is the little movie which explains how the ILC will work. Not this one, no. You touched nothing. I haven't touched anything yet. You're going wrong? Okay, yes, it's this.
Yeah, the only way to do this appears to be to not do anything. This works perfectly.
Oh, there we are. That sounds good. Okay, thank you. Okay, here we go. So this little movie explains how the ILC will work. So you probably have to click on this little thing. Here. Yes. Oh, sorry. Okay. So, can you all see? Yes. So ILC, so here we see. So it will be buried around 100 meters underground in the beautiful country in north of Japan, I will tell you later. And this is where they will collide at the end. This is where the electrons are produced. And they go around this little ring here, a few kilometers long, and get sent to the other end. And here they're used to make positrons, anti-electrons. They go around, sent all the way over here. Then they meet in the center where they collisions and occasionally produce Higgs particles, which we then look at using these big cameras. We call them detectors, particle detectors, basically a big digital camera which records what happens in the collisions. So if we zoom out a little bit, we can see more in detail what's really going on inside. So we go to the end of the tunnel over here. We go into the tunnel, and you see this yellow tube. The yellow tube, inside the yellow tube is minus 270 degrees Celsius. Outside is room temperature, so everything is superconducting inside. And when we look inside, what's inside? Inside are the things which push the electrons and positrons to make them go at the high energies we want. We look inside, we have these little lumpy structures you can see. And inside there we put some electric wave which pushes the electrons and accelerates them as they go through this structure, always like surfing on a wave, for example, until they come near to the end of this part, which will be soon. They go faster and faster and faster. And then at the end they are focused down to a very small spot where they then will collide in the middle of this big digital camera, which we use to detect what was going on in each of these collisions. Okay, so there you can see the whole 20-kilometer length of it. Okay, thank you. Let's go back. So let's go back a few slides. Okay, I can do that. Okay. Control L. Oh, with that? Okay, great. Thank you. So we hope to build this machine, this ILC, in Japan. We found a very nice place up in Iwate Prefecture in the Tohoku region of Japan. And this will be built as an international project. So there'll be many contributions from countries all around the world. At the moment, researchers from the Americas, from Europe, from Asia are all working together on this project and all pushing to realize it. We have strong support from politicians in Japan, both national ones and local ones, from local governments in Japan. In particular, I want to thank our Iwate people who are sitting over there, give us a wave. And from also industry, who will help us to build this machine, from scientists all around the world who want to use it to measure the Higgs boson and other processes. Yes. So the government in Japan is at the moment studying we asked them, can we please get some money to build this? So they're studying this, and we hope to get an answer very soon, in the next month or so, as whether we can go ahead and build this machine to realize all our dreams. Well, not all of our dreams, but some of them. So if you're interested, I point you towards this ILC supporters. So this is a grassroots general public group of people who are supporting the ILC, who are enthusiastic about the science and the internationalization this can give to Japan and things like this. So if you're interested in learning more information, check them out. You can sign up as a supporter of ILC to make your voice heard. As you see, now we have almost 300,000 people who've signed up for this. So, in conclusion, I think we understand surprisingly or depressingly little about our universe. In this room, we understand only 5% of it. And I think ILC will be a great tool to help us to get to know this 95% a little better, at least. Okay, thanks.
So now we have a few minutes, only a few, for Q&A. You will now stand trial amongst a bunch of non-experts.
But there are a couple rules.
Yes, indeed. Keep your questions relatively short. If you go over 1 minute, I will beat you. Beyond that, I believe that's it. So yes, that's more or less the deal. Please be at least concise in your questions. Go ahead.
Actually, my question is kind of big. So what does it mean for a particle like Higgs to exist by itself? What I mean, you said Higgs gives mass to other particles. So if there are other particles and they have mass, then it means that Higgs is around to give it to them.
Well,
so
it means something is around. It's not necessarily Higgs, but now we've discovered it probably is Higgs. Yes.
Well, assuming the definition is the theory is correct, if there is a particle with a mass, there must be Higgs near it. So why don't we just see it around? Why we need to collide those like other particles, and see the standalone Higgs, and if it's standalone, doesn't it give like mass to anybody? What does it do like out there?
So I think I understood your question. So many of the particles I showed in this table don't live for very long, Higgs included. Most of them decay immediately. So to have a standalone Higgs sitting by itself, it will never, well, it will sit there for 10 to the minus 24 seconds or something, but then will immediately decay into something else. And you said we should assume the theory is correct. This is exactly what, I'm an experimentalist. Our job is exactly to prove the theories are incorrect and to find the correct one behind it. I don't know if that answered your...
Okay, so the Higgs that is produced in the collider is like, has more energy, so it lives longer or something like that?
It doesn't live long, no, it decays immediately, also in the collider. But we can see what it decays into, and we can reconstruct that it was there.
So just now when you said that you hit a lot of positrons and electrons to just get 1 Higgs per hour, is it just because the method is so inefficient, or is it just because you need a lot of energy to produce 1 Higgs?
So the we have, in each collision we have enough energy to produce a Higgs. One problem is the electron is so small, it's almost infinitely small. So to get them to really collide together, to get them to meet each other after 10 kilometers each way is rather difficult. That's one problem. But then also there's the electron doesn't interact so strongly with the Higgs, for example. So it's difficult. It prefers to make other particles than the Higgs. So only rarely we are lucky. There's some probability that we will produce a Higgs, and it's rather small.
Do you think that this method is like the most efficient? That's why, you want people to also support Japan to, adapt this.
No, so, for example, Higgs is also made now with in Geneva with proton-proton collisions, for example, which is maybe more efficient, but when you produce a Higgs in proton-proton collision, you produce a Higgs plus an enormous amount of junk all around it, and you have to pick up in a little haystack where this Higgs particle was. Here we produce 1 Higgs particle, nice and clean. We can see it beautifully. We can measure it precisely. We can really see it clearly.
Okay. A quick question. So, like, but nasty questions. I'm totally a supporter. How much? And it must be super sensitive equipment. Earthquakes, right? So.
Okay. So how much money you mean, or how much time? So how much money? So it would take. Okay, so it will cost about the same as LHC. What does that mean? That means, so it will take around, okay, first I will give the good news. It takes around 10 years to build, and then it will be used for at least 20 years or 30 years in the future. So this is a 50-year-long project. And to build it initially costs billions of dollars, maybe around $7 billion, something like that. And that's exactly because it's very sensitive equipment. So the reason, in fact, the reason we build it in Kitakami, in this northern region of Japan, is there's basically a big piece of granite, 50 kilometers long, in which we put it. So everything will move together beautifully. It's all kept fixed together by this enormous mountain range.
So thanks for the great presentation. So let's imagine this is working, and you have a system which is producing 1 Higgs boson per hour, and it lasts for 10 to the minus 24 seconds. What do you actually hope that you can figure out about the Higgs boson from this experiment?
At some point I gave some list of questions we have, for example.
Sure,
about
questions, but I mean...
So we hope to get the answers to these questions.
Okay, that's what you hope to get. So.
Yes. Well, we will get the answer to these questions. Because we know the Higgs exists, and we know how to make these measurements.
Yeah.
We don't know what the answer will be, but we will get the answers to the questions.
Okay.
But we don't know what the answers will be.
Okay, thank you.
Hi. So, I've heard that there are other countries vying for the ILC. Is there still competition for this, and what's the situation?
So the ILC is sort of, since it's been around a long time, so first we proposed a very similar machine in Germany. That was maybe 15 years ago or something like that. That didn't fly for political reasons or whatever. The financial crash happened at more or less exactly the same time, so that was rather unfortunate. Then the US tried to get this going, but it's never difficult to get budget for big science in the US, so that also went. So at the moment, for ILC, Japan we are all getting behind Japan to host ILC now. There are other similar projects being proposed. There's one being proposed in China. It's not the same. For example, it's a ring-like accelerator, which will also be used to look for the Higgs. So that's maybe a competing project. But for ILC itself, Japan is, we put all our cards in Japan. I think you have one behind you.
You partially answered this, but how is this different from the CERN? Because of circular versus linear?
So now this big difference is between colliding protons and antiprotons. So let me go back a little bit to one of my first. Okay, so we collide one of these guys at CERN. They collide one of these guys with two of these guys together, and these guys have 3 quarks together plus a lot of glue to keep it all together. So so you can imagine if you, some people give the analogy, this is like a cherry pie, for example. This is like splashing 2 cherry pies or 2 custard pies together. Occasionally one of the little cherry pips stones hits one of the other cherry stones and makes something interesting, but together with that you get enormous amount of pastry and jam and messy stuff. When we do electrons, we have only the cherry pips themselves. We don't get this enormous junk all around it. That makes it possible to see much more clearly what's going on.
Could they collide electrons if they wanted at CERN?
So this machine, the tunnel where they now have the so-called LHC, this proton collider, used to be used for electrons. I used to work on that, particularly when I was doing my PhD. But the problem is when you have an electron and you bend it around the corner, it loses a lot of energy. When you accelerate an electron, or any particle, it loses a lot of energy. Because the electron's very light, when you try to make it go around the corner, it loses almost all of its energy just going around the ring at one time. So there are proposals to make an enormous ring accelerator, which can do this Higgs stuff, like this one in China, but this has to be 100 kilometers long, something like this. Okay, let's leave it there. Linear, you don't have this problem of bending the particles, so they don't lose the energy, so it's much more energy efficient in this way.
So I have a related but very basic question. You call it linear collider, but you showed the video and it looks like a circle to me. What does it mean that it's a linear collider?
So there are circles in it, yes. I will try to, we have a picture here, right? Somewhere. So linear, so it has this circle here. When the electrons are going around here, they're very slow, well, relatively slow, low energy. So then they don't lose so much energy when they go around the ring. The linear part is this part here, which is where the electron starts off very slow and accelerates to a very high energy. So this is what we call the linear part. This is the accelerator part, in fact, this box, this long tube.
We have time for 2 more questions. 2 more questions. Any more can be done during the break.
How do you make anti-electrons?
Nice question.
Sorry, I couldn't hear the question. Could you repeat that, please?
How do you make anti-electrons?
Okay. That's fine.
You weren't listening at the very beginning of this talk, young man. So you start off with some energy, and you turn it into an electron and an anti-electron. You take a photon, you bang it into a piece of lead or something, and then out comes an electron and anti-electron. Simple. You collect them using magnets and so on. So you use magnets to steer where they want to go, where you want them to go. They come out with some speed, and you can use magnets to steer them, down the pipe.
So with the ILC, do you expect to generate some other particles along with Higgs, or just like dark matter or something?
So this will be super exciting. So there are also possibilities that, we know we will see the Higgs. This is sort of guaranteed. But maybe there'll be other particles we will find too, new particles. Many people, many theorists predict new particles. I don't believe any of their theories, but maybe one of them is correct. And we have, yes, so there's definitely a possibility we will find new particles.
Do you have like expectation what you might find along with Higgs?
It depends which theorist you believe. I believe none of them, so I would be great to find something, but I can't promise it. And if we do find something, I'm not going to put any money on which one it is at this point.
Last question. Don, it's up to you. You want to give it? Nope, there it is. There it is. Oh no, give it to him. Yep.
Hi, I was wondering why Japan? Because Japan, out of all places, is a tricky place to have sensitive instruments. Is it the money?
So Japan is, I would say more than money, it's the political will, the political enthusiasm for research, for fundamental research, which is sadly lacking in many other countries in the world, where such things are not so valued. That's one part. Also, other parts of the world already have big fundamental science projects. So in Europe, there's already this big Hadron Collider. So it's difficult to imagine how they can afford to have two such enormous things. US is also developing one to look at neutrino particles, also very big experiments. So it's difficult to imagine one region making two such enormous things. And Japan doesn't have a really big one at the moment.
I think we can call it there. I'm gonna, I really want to push you into the lights because you're really off the light. So we can see everybody on the stream. Thank you very much for coming out for this, for Irma here. Thank you very much. Thank you. Scientists and theatrics. Theatric folks always look for their light. Scientists always look for the dark shit. Two more talks coming up. We're going to have a break until 9:05. It is currently 8:46 right now, which gives you how many minutes? You know how to count. We do have flyers up front for an art exhibit thing happening from some Boston awesome cool thing called Bad Art. It's pretty awesome. Go check out the front. But anyway, grab a drink, hang out, grab some food, talk to us. We're around. If you want to do a talk, come talk to us about it. And Andrew's going to take photos of me with a really... I'm so sorry. I'm standing straight here. Anyway, 20-minute break. Go! Thank you. We'll see you back at 9:05. Thank you for that tepid applause.
John. You know what time it is?
Is it time to mic up the second speaker?
It's time to get
To mic up the second speaker? To mic up the second speaker first?
No, we're doing some promo.
I can't, what now?
Promo.
I can't promo shit. I can't even talk right now. Like, people are just so busy. Like, they're just so I can't promo nothing, man. I'm just saying, I don't know what you're talking about. Is everything cool here? Seems to be. Yeah. Everybody's calmed down now, which is nice. You know, quiet y'all asses.
John is doing it.
Yeah, I'm doing everything. Hey, Joan. I see you crouching here. What are you doing? Have you mic'd up the second speaker yet? You have? Yes. Oh my God, you're so good at this. Is the power on the speaker? Yes. Oh my God.
It's like you've never done this before, Joan.
It's like she's actually done this several times before. She's super helpful. Joan is awesome. Oh shit. Is there light? Oh, I feel so warm. Well, firstly, peace and long life. No response? Come on, goddammit. It's live long and prosper, dicks. Thank you. No, it's cool. You have to just put the things together and yeah. So no Trek fans here tonight then? None? Any? Trek fans? Any? Okay, so favorite captains. Picard? Hang on, Kirk? Whoa, eat it, please. Eat terrible things.
Crunch?
So Picard, Kirk, Cisco? I'm Cisco, by the way. Yeah, that's right. Respect! What's happening? Come talk to me later. Janeway? Any Janeways? Happy, much respect, but also wrong. Lorca. He is the shit! Oh, I love Lorca. Watch Discovery if you haven't yet. It's CBS and they have it behind a paywall, but if you're in Japan, it's on Netflix because licensing. Shit, what are we talking about? We got the next thing we're talking about, which is the second thing, which is speakers. What is this? This is not what I'm talking about. I'm talking about something really fun, which is the place that has provided us this awesome venue. Wait, this is a video? Do I have to talk over this? Is this so loud? I just want to talk over this. Okay, so children are starving, per Andrew. But this place is called Gaiax, Nagatacho Grid. Awesome company. They provide us this space for free. If you register via one of the links on one of those postcardy things over there next to my station / the bar, you can hang out here for free. Coffee is free. Space is free. Just hang out, collaborate, co-work, do cool stuff. Sounds too good to be true. It probably is, but we haven't noticed yet, so it's obviously fine. They do yoga nights, movie nights. Yeah, I skipped over yoga nights really fast in case you don't like that kind of thing. I still actually do yoga, which I'm really embarrassed to say. Movie nights are awesome. Being here is cool. You can use the space free of charge, unless you're like really making tons of money, in which case you're abusing the system, so stop doing that. People are happy, obviously, and they're doing that near here. Obviously, it's happening nearby. Yes. That's here. That's upstairs. Yes, people are happy. That guy has a 70D. Kai has a 70D. We have a 70D together, but it's really mine.
Moving on.
Yes, I'm going to move on when the damn video ends, okay? I'm just waiting on this.
We didn't choose it.
Yeah, we didn't choose nothing. Don't do that. Whoever just said that, I'm going to break you. This is not the video.
It's the same video. Can we just like move on from the video?
Are we allowed to do
help? Yes, help.
Parkour.
Okay. You can do Andrew. You can do parkour on the roof. You're wearing a kilt. Okay, so I got this. So we do love this place. Seriously, their videos are long and yogi, which is not my taste, but we do love this place. They give us this space for free, and you can come use this place for free as well. So come hang out if you want to be part of the community. Doesn't cost you anything. Go over there. There's a link to where you can sign up, talk to us. You can talk to me if you want to. We know how it all works. So beyond that, we have some very dear friends over at this excellent event called Space Cafe. It's run here in Tokyo and in Washington, D.C., which is two very different places. The next one is happening at Good Heavens Bar, Grill, and Emporium of Comedy on November 13th at 8 p.m. It's in Shimokitazawa. We used to actually have our venue there for those who have been with us for a while. I know at least 3 of you have been there so far, because I know you people. They're talking about awesome space stuff, and they always do. It's a great place to be. If you're in Shimokita on Tuesday nights, it's almost always on a Tuesday night, go hang out. Next thing, please. Thank you, Desu. Don, human? Yes! This is my thing. If you were here last time, you recall that I mentioned we're doing a gaming group here in Tokyo. There are very few kind of like casual hangout, play video games. We all have really small apartments. We don't like invite friends over to hang out because we're first of all antisocial. Second of all, our homes are very small. So now we are doing stuff at places like bars and cafes and bars and people's houses occasionally. It's called Tokyo Casual Gaming Encounters. If you are a video gamer of any sort, please feel free to look this thing up. I encourage you to pull your phones out and do it now if you're a gamer type, or come talk to me after, or email me at that very hard to read email address down at the bottom. But come see me, seriously, if you want to hang out and play video games. We hang out once a month, safe environment, no harassment allowed. I will physically beat human beings to death if it happens. It's happened in other communities, okay? I'm just trying to, you know, be a decent... Well, but I know it perpetuates, but if they're dead... It is Henry. Longtime Nerd Nite supporter Henry, who is a boss guy. This is actually my apartment in Yokohama. Yeah, he's having a really good time playing, I guess, something with guns. It is something with guns, actually. I'm well aware of what it is. Yeah, so we're going to get together a group stream and stuff, video game stuff. It's going to be a lot of fun. So that's enough for that. Don, I think you can probably push button on the next thing, which is scroll, 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 scroll. Yes! We give money! And you get a grant, and you get a grant, and you get a grant. I know that joke is so overused. But Oprah is great, so we love to use it anyway. If you are doing a fantastically interesting, researchy, sciencey project, come talk to us. We do give out grants. We will want you to present here at Nerd Nite. We'll want it to have some bearing on what's happening here in Japan and some other stuff in general, not necessarily, but it's very much preferred if it's close to home. Those are sort of the general requirements. And Amanda wants to add something.
Here's an example of what Nerd Nite in Seattle has given a grant for to create virtual reality art, a little book, and a USB drive. So that's just one idea of something that has been funded with Nerd Nite grant money.
We give you cool money to do cool stuff, so be cool, okay? If you do want to apply for a grant, come talk to Amanda right here. She's going to stand up and turn around and look at you all and be really shy. Nope, she's going to stand up.
No, I'm going to say, point to this QR code
wow.
behind John's butt right here.
Who uses, my butt is like over here. You mean my shadow butt? No, so take a picture of that later if you want to apply for a grant.
Oh, and?
I see one person with the camera out. Thank you, appreciate it. Yeah, come see us.
And we actually are giving the first grant. Somebody has applied, and we're going to have that person come out in 2019 to talk about their study. So, yeah.
Yeah, we want to support the community doing interesting research and interesting products. So projects, excuse me. We're not that commercial. So come out and talk to us if you have anything cool you want to do and you're looking for a grant for. Okay? We don't have a ton of cash, but if you're doing something interesting, we'll try and help you out. All right, I believe that covers most of the topics we're talking about. Yeah, I believe that's it. So where's Thomas at? He's right there. Is that him? Is he ready to go? He's working on his laptop, but he's not going to stay right there because he's going to change his laptop over and get the slides ready. And when he has the slides ready, he's going to come over here in the light because the stream is right there. And it's pointing in this direction. Make sure everything's good to go. That's right. So again, Cisco Captain Cisco fans, come talk to me afterward. Just saying. You ready to go?
Maybe.
All right, so I will intro you now that you are ready to begin your speak. Thomas is a British neuroscientist currently working in the Center for Brain Science at Riken. He likes drums, guitars, beer, and brains, which two of those things.
I like at least one of those things, yeah.
Well, then he likes beer.
Yeah, that's the one.
Yes, he does, Your Honor.
Yeah.
Now, yes, political joke, timing. Now he's going to tell us about the biotechnology that makes brains glow. Thomas, take it away.
Thank you. So,
yes.
Yeah. So I think I've given loads of talks hungover, but I don't think I've ever given one drunk. So it might be a shambles. So tonight is it's a very scientific night, right? So there's like there's three scientists. There's a mathematician, a physicist, and a biologist. So we're kind of covering the spectrum. So, you know, the mathematicians, it's a spectrum, right? The mathematicians, logic, philosophy, and then maths kind of sets the scope for the universe. Physicists are looking at the really small parts, and then sometimes you get molecules, which is sort of the next level up. I don't mean any offense by any of this is all positive. And then sometimes, though, the chemists, sometimes molecules self-organize. And then that's when you get life, right? So biology. And then sometimes life starts to think. And so this is neuroscience. So this is where the sort of level I'm trying to be at. And so we're trying to understand this. What is this? It's a brain. Very good. Yeah. So whose brain is this? It's not mine. So you have to leave now. Whose brain is this? What kind of individual? This is a human brain, right? So how is this person doing? What? They're super dead, right? So this is a fixed human brain that's been removed from the skull. This person is dead. But we want to understand this object, okay? But there's loads of things that come out of this object. I mean, these are all common everyday things, right, that we're all very interested in. Thoughts, feelings, emotion, all of art, all science, all, all maths. Well, maybe maths is more fundamental than brains. The word brain comes out of the brain, like, right? I would say soul religion comes out of the brain. It's controversial. And there's loads of problems with studying that object. Okay, let's start the simple stuff. It's in the head, right? So your brain is inside your skull. It's a hard structure. It's not very transparent. It's good for us when we're walking around and falling over, but it's a problem for a neuroscientist. It's a complicated structure. I'm going to come back to this. The consistency is something like blancmange, okay? So if you get a spoon, you can just scoop the brain out, right? Or maybe in Japan, purin is a better example. It's super easily damaged. You know, anything noxious or toxic or, damaging might damage the brain, right? So to study it is a massive pain in the ass, basically. So we would like to be able to see the individual components of this object. We'd like to be able to see how they're connected and what they're doing. That's the goal of a neuroscientist. So this is the same person's brain chopped up. So if they weren't dead before, they're definitely like dead now, right? We can see a bit of structure in this object, right? You can see some stuff going on, right? Holes in the middle, some wibbly bits around the outside. There's some white part here, there's some gray part here. But we can't see the individual components inside this object, right? You know how big a brain is. It's a mess. We want to kind of get deeper. This is a big problem for neuroscientists. The brain is essentially colorless. It's kind of a gray blob. So what we'd like to be able to do to start off with, to solve this problem of understanding it, is highlighting individual brain cells, some way of doing this. Neuroscience is a new field. About 150 years ago, it's relatively new as a science. Neuroscience started with these two guys. So Golgi is an Italian guy, a dead Italian guy, and Cajal is a Spanish guy. And they together essentially started neuroscience. So Golgi developed a way, if you stain a piece of brain tissue with some chemicals, you will randomly pick out a few brain cells. So they'll go very black in this object. And Cajal took these stained samples, and with a lot of patience, he sat down with a microscope, a piece of paper, and a pen, and drew what he could see. And he drew stuff like this. And this is a brain cell. This is a neuron. Okay? So this is the first time humans basically saw what was inside the brain, the actual individual components that build up the brain. And they won a Nobel Prize for this. So, it's a massive breakthrough. This is the origin of neuroscience, essentially. Apparently, they didn't like each other, and they disagreed a lot, but they won the Nobel Prize together for this. So this is really good, right? We can see now inside this object, we can see individual neurons, but there's some problems. It's a snapshot in time. It's not a movie, right? It's just an individual, it's a photo of the brain. And what's another problem with this? It's super dead, right? Like that brain tissue had to be soaked in chemicals, many things had to happen to it. It's a dead snapshot of a brain. So it's massive progress, it gets you a Nobel, really good. But we'd like to get a bit further into this, right? Get, more detail. So another way of doing it, a very common way of doing it, is you take a needle, or a small glass needle, which is hollow, some dye and a steady hand, and a Scottish guy in this case. And you stick the needle. This is now a cover slip with lots of neurons growing on it. So we're looking down from above. And this is a needle or an electrode coming. And so we can stick a tiny hole in this brain cell and fill it with a dye. So now we can see this neuron, okay? So it's this incredible structure, right? And so we can start to see like the sub-compartments of this cell. This big blob in the middle is the cell body. This is where the nucleus is. This is where the DNA is. This is where the instructions to make neurons are. Out here, we've got all these projections, which are dendrites. So this is where information is arriving onto this neuron. Okay? These are the listening parts. And up here is an axon. This is the output part of the cell. So in reality, when I took this picture, the axon actually like continues miles off the screen. The axon is very, very long. So information is coming in to the dendrites, it's being filtered, going through the cell body, and then when the neuron wants to talk to other people, it's going out through the axon. And this is living when we took this picture. So we've solved one of the problems with it being dead, right? We took a picture when it was still alive. And it also allows us to record activity. So instead of just taking a picture, like a snapshot, we can actually hook this up to an amplifier. So it's not a Marshall stack, but you know, it's an amplifier that will amplify the very tiny electrical signals in the neuron. And then we can record the activity of the neuron. It's like a heartbeat. Every time the neuron is active, there's a beep, beep. So this is great, right? We've got the morphology, we've got the shape, and we've also got the activity. This also won a Nobel Prize, this technique. So this is now in like 1950s or something. So just sticking a needle in a neuron wins you a Nobel Prize 70 years ago. So easy for these guys, right? So this is really good. We can see the shape of the neuron. We can also see the activity of the neuron, but still lots and lots of problems. This was in a cover slip, so it's not like a real brain. It's a very simple preparation. This is very time-consuming, so a single neuron, and it's very invasive. So we have to literally stick a huge needle into the cell. So, whoa. Can we do this a better way? So the dye that I was using in that experiment has this property of fluorescence. You shine light on this dye and it will emit light of a different color. So for example, if you shine blue light on this dye, it will emit green light. Okay, this is very useful. And it turns out some animals are naturally fluorescent. So some animals have this property. So this is a famous example of a jellyfish that if you shine the right color of light on it, part of the jellyfish fluoresces. And this is GFP. This is the most famous protein probably in biotechnology. So this is green fluorescent protein. So this was isolated about, I guess, 20, 30 years ago, and it was so successful, this fluorescent protein, it spread worldwide. Like nearly every biological lab is going to use this. So what do you think the discoverers got? They didn't get Bupkis. What's after Bupkis? Yeah, of course they got another Nobel, right? So the people who developed GFP won another Nobel. This is a huge breakthrough for neuroscience per se, but for biology. So now let's get GFP, this fluorescent protein, and just put it into some neurons in a slice of brain and take a picture or a movie. So if you imagine a slice of brain, it's essentially like sashimi. It's living, right? So it's a very, very thin slice of brain, and inside the brain there's many, or inside the slice there's many, many, many neurons. And some of the neurons have GFP in them. So if we scan our camera or our microscope through the slice, we'll see this kind of thing. So we see all these wiggly bits, wiggly, 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 and then down here we'll see some blobs. These are the cell bodies, and then lots more wiggly bits here. So if we just take all those pictures and collapse them, we get this picture, okay? So this is about 0.6 millimeters. So these are small objects. They're nowhere near as small as molecules, but these are small objects, okay? So in this picture, we've got two neurons and a third one down here. But in reality, this picture is full of neurons, but we only see the ones with GFP inside of them, right? The other ones are invisible. And this is a three-dimensional structure, right? So if we take all the images I showed you at the beginning and turn them into sort of a rotating movie, you can see it's not flat on a cover slip. This is a three-dimensional structure. We've got the cell bodies here. These are all the dendrites projecting out. And then these really, really thin wiggly are the axons, okay? This is living. When I took this, it was alive, okay? The sample was alive. So it's great, right? We can actually look at living brain neurons in a kind of slice of brain. We've made a lot of progress compared to where we started. So that was GFP, green fluorescent protein, one color. How about lots of colors? So scientists took GFP and mutated it, and also, probably not scientists, but adventurous people went into the oceans and to the jungles and found other fluorescent organisms and found out how to code for their proteins. And we've got this now palette of fluorescent proteins. And this image is quite old, so this is kind of out of date. We've got blue, yellow, all the way to green, all the way to far red. So we've got multiple colors now we can use in living tissue to look at neurons. So the first thing you could do if you're an artist, and I guess like a biochemist or a bacteria guy, is paint. So this is a plate of agar where people have painted someone. I don't know actually the source of this. Someone has painted with bacteria. So each color is a different species of bacteria or a different bacteria that's expressing a different fluorescent protein. They've painted, I guess it's a setting sun with the green flash. It's maybe Hawaii or somewhere, right? So this is amazing. This is alive, right? This is living when they took the picture. But in my context, we might put them into neurons. So the first thing we could do is just put some in red in some neurons, put some green in other neurons. And so here's an example of an axon of a green neuron and a dendrite of a red neuron. And we're starting to see that maybe here, maybe they're connecting, right? So now it's useful to have multiple colors. Maybe we can see connections. Another thing to do with this is mix colors. So just green in one cell and red in another cell. We can mix them. So say we start out with 3 colors, blue, green, red. So anyone know where this meme comes from? Where? Yeah, oh, the internet. Oh, come on, yeah, okay. Yeah, the internet, yeah. If you put these into a brain sample, you would definitely have some blue neurons, right? And some green neurons and some red neurons. But if each cell takes up a random mixture of the 3, you'd actually end up with something like this. So you have blue neurons, but you also might have some neurons that have a lot of green or yellow and red, which would be light green. And you might also have some neurons which have blue and a bit of red, which would be purple. And if you do this, you end up with this, right? So this is a rainbow mouse. So neuroscientists are like really funny people, so rainbow, brainbow, right? So this is a real... But, you know, it's a good name, right? That's probably quite easy to think of, but this is an awesome image. So this is a real brain. This isn't like a fake brain. This isn't like a super manipulated image. This is a little chunk of a rainbow mouse. So each one of these blobs is a cell body of a single neuron. Up here, this kind of mess is all the dendrites. And down, this is all the axons, okay? So we're seeing all these projections. We're starting to get an idea of how complicated this structure is. Like, it's really fucking complicated, right? Like, after watching the first talk, I thought, actually, that's more complicated, but everything's quite complicated. But brains are like common, right? You don't need to find one every hour with a linear collider. There's loads of brains in this room. You've all got one of these in your head, right? They're super common, these structures, but they're super complicated. So human brain has something like 100 billion neurons in it, in your head. 100 billion, approximately, give or take 5 or 10. So let's step back a little bit and just look at a single neuron, because that is too complicated. So one neuron that's expressing GFP. So here's the cell body, and here are all the dendrites, and let's zoom in a little bit on a portion of it and see if we can see some more detail. So if we zoom in on this region of dendrites here, and then we make it picture a bit better, we start to see there is additional detail, like in this, right? Let's zoom in again. So this is a dendrite, and we're starting to see some like wiggly bits on the dendrite, or some projections, okay? So this is actually what I work on. So my like tiny portion of neuroscience are these objects. So this is a prettier picture. It's a 3-dimensional structure, but this movie doesn't work. So let's let it not work. So it's a very long tube, it's a very thin, long tube with loads of blobs sticking out of it. And these blobs are dendritic spines. So this is the dendrite, and these are the dendritic spines. And these are where other neurons are talking to this neuron. So each of these blobs here has another neuron talking to it, okay? And then this is actually an axon of another neuron in this image. So this is coming in and it's contacting this spine here. Where axons meet spines, that's where synapses are, or synapses if you're from the UK. So this is the connections between neurons, synapses. And this picture probably has maybe 60 or 70 synapses on it. The size of one of these is about 1,000th of a millimeter. So these are small objects, right? These are very, very small objects. You need a microscope to see them. And if you look at like a whole neuron, they're covered in spines. So if you zoom in here or here or here or here, and you would find this, okay? You find all these projections all over the neuron. So this is a single neuron. So it's something like, in a human brain, 100 trillion to 1,000 trillion synapses, the connections between the brains, the neurons. So this is the object we'd like to understand. Lots and lots of neurons and lots of connections, and how the connections change in strength over time. This is where your memory is. This is where you are. This is where your reality is encoded, essentially. And within each neuron, there's loads and loads of proteins. There's loads of stuff happening within a single cell, or even within a single synapse. But again, there's no color. If you just take a picture of it, it's just a gray goop. So can we use GFP here? So if we zoom in to a little region of the cell, these are loads of different proteins in the cell, but we can't see any contrast. We can't identify a single protein. So if we just stick GFP onto a protein we're interested in, wherever that protein is, there will now be a green signal in our cell. So this is super useful. This is probably where GFP is used most in biology. So here's a real example. This is a neuron which is full of a red protein, so it fills the whole cell. So these are all the dendrites, these are the spines. And then here is another protein in the same cell which has been tagged with GFP. So wherever that protein is, we'll get a GFP signal. And you can see, like, this guy is obviously targeted to specific regions within the neuron, right? So this is actually a synaptic protein. This is involved in how synapses talk to one another. And this is extremely useful. So we could watch, where does it move? Does it change? When you learn something new, do the levels move up or down? Stuff like that. So far, we're doing really well. The timer never got started, so I have no idea what time it is, but I think it's maybe okay, right? So far, we've seen the shape of neurons, right? We've also seen maybe the connections and the fine scale shape of neurons. And we can also localize proteins within neurons, or any cell, really. How about activity? How about when neurons are talking to one another? This is, you really need this to understand the brain. You need to see when neurons are active. So, to skip 20 slides, take my word for it. When neurons are active, the level of ions inside them change, and calcium increases. So whenever a neuron is active, whenever it talks to someone else, the levels of calcium increase. Whenever a synapse is active, the levels of calcium increase. So we can take advantage of that. So the way people took advantage of this is they took GFP and they cut it in half. So they circularly permutated it. I don't know why this is covered with blood. I think it's, I can't remember where I got it from. So you cut GFP in half, so now it doesn't fluoresce, it doesn't work anymore. And you stick another protein to GFP, which likes calcium. So this protein will stick to calcium, okay? So you've created like a Frankenstein protein, right? Like a fake protein, but it works. So whenever this protein, which is not fluorescent, the GFP halves are separated, whenever it binds to calcium, it will change shape slightly, and it will bring the two halves of GFP back together. And this is fluorescent, okay? No calcium, no fluorescence. With calcium, bright green neuron, and it will emit photons of light, right? So this allows us basically to track calcium levels in the cell. Whenever calcium increases, it'll go bright green. If there's no calcium, it'll be dark. So this is now a neuron growing on a cover slip. Here's the cell body, here are the dendrites, and can you see the little flashes? I'm not sure, I think the movie's okay, right? Can you see little flashes? Yeah? So every time you see a little flash, that's another neuron talking to this one. We're watching this neuron listen, essentially. So if we zoom in here, here's a dendrite. These are all these dendritic spines that I talked about previously. Every time you see a flash, another neuron has talked to this one. It's sent a message. So now we're actually monitoring activity in real time. We can watch the neuron receive information, and we can also watch the neuron output information. So this is cool, right? No, okay, this isn't cool. Okay, we can do better. That's very depressing. So that was in a single neuron growing on a cover slip. So that was a super artificial system. But you know, it's still quite difficult. How about like lots of neurons at the same time? Or how about a whole brain? So for legal reasons, we can't do this to humans, unfortunately, but we can do it to fish. So this is a picture of a fish brain, a very small transparent fish called a zebrafish. And this picture, we're looking down from above, okay? So these are the eyes. This is the sort of main brain, and this is the spinal cord. And every neuron in this fish's brain has this calcium-sensitive GFP in it. So whenever the neuron is active, you see a flash. Okay, so this is now, A, it's good. Here's the time. Okay, so it's sped up, right? A, it's good we can do this, but B, this is also one of the big challenges of neuroscience now, that we're generating these huge data sets. And we're trying to figure out, what does it mean? You can make a really cool movie, right? But what does it mean? How do you extract the data? You know, this is where the physicists and the computational neuroscientists are really, you know, super involved. So this is a fish brain. Who cares about fish? Hands up. Who cares about mice? More people care about, it's about 50-50, right? So in reality, a mouse brain is not so transparent, which is a problem. But you can, people have developed techniques now to treat mouse brains or human brains, if you're allowed to, with chemicals to make them transparent. So the brain is still there, but you can now see straight through it, okay? So this is a dead brain, but we can now start to see deeper into it. So this is really useful. And so this is actually from the paper. This is a sort of movie of someone imaging in this brain. They can see this is the top of the brain. They can see all the neurons in the top, and they can also see all the neurons in the deep structures. So for a neuroscientist, this is a big breakthrough. Also, the guy who developed this hasn't won a Nobel, but probably will win one for something else. So this is 4.5. And also, so this is actually an example from America, but there's a Japanese group doing exactly the same thing, making brains transparent. So this is the end, basically. Rampant speculation. Up until this point, it's been pretty solid scientifically. There's lots of transparent creatures out there. So this is I think this is a glass frog. This is some kind of octopus, which is just transparent. It's wandering around doing its life, and it's already transparent. So if we could figure out how to take the proteins out of these guys that make them transparent and put them into mice, then we could image very, very deep into mice brains. So, and that will give us some idea of how human brains are working. It's going to be illegal in humans for a long time. So, conclusions. Well, depends which country you're in, right? Whoever said that. But yeah, you know, brain is very complicated. Donald Trump is a fucking idiot. And, you know, easy audience, right? Scientists. Yeah. A round of applause. Oh, yeah. GFP and other fluorescent proteins are helping us map and understand the brain. So don't take a picture of that, Amanda. Yeah. So this is an image that was drawn by Cajal using the Golgi stain in 1911 of the hippocampus, which is a brain region involved in memory. And then this is an image taken about 10 years ago using the Brainbow mouse. So you can see the sort of how technology or this biotechnology has developed. We're starting to see more and more details. And that's it. References, anyone want to join a band or start a band? Thank you very much. Any questions?
Yeah. The statements herein do not represent appropriately necessarily the blah 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 political statement.
I'm very curious. So when you said that if a protein is like separated and then you try to glue them again together with some calcium or something that likes calcium, does it only work for GFP or it works for other proteins?
Essentially, you have to make them bespoke, right? So it doesn't always work. You have to try and make it. So people have tried to do it for other, like sodium, which is much harder, but would be much better. So I use the most kind of obvious choice, but yes, in theory, for anything.
Okay, and then if you get them back together, do you get back the original properties?
In this case, in the case of GFP, the only property we care about is does it emit a photon or not, right? Is it fluorescent? So any of these manipulations I've showed you, I said it was better, like not sticking a needle in the cell, but every time we do something, we do something. So there are definitely downsides. We may have changed the protein's behavior, right? But in that case, with the GFP calcium sensor, we only care if it's binding calcium and emitting light. But probably, yes, it has changed the properties, yeah.
But it doesn't matter, okay.
No, it does matter. It totally matters. But there's limitations to everything we try and do, right? Yeah.
I see.
Like filling a neuron with GFP, maybe the neuron doesn't want to be full of GFP, right? So everything you do has limitations. It may change the behavior of the object, yeah, for sure.
Hi. So I was wondering, how do you actually get mouse neurons to express different proteins, and what does it mean when the neurons are different colors? Are they different types of neurons, or it's completely random?
So, first question, so there's many ways. You need to take the DNA that codes for the protein and get it into the cell. And then the cell will take that DNA, read it, and make the protein. So the most common way is you get a virus. You engineer a virus. You put your DNA in the virus will naturally infect neurons and then make the protein you want to do it. The way I do it is with actually a gun. So it's a shotgun. So I take gold particles, tiny ones, and I stick DNA to them, and then I use helium to fire the gold particles into the brain slice. If you're very lucky, the gold particle sticks in a cell and doesn't kill it, and then the cell makes the DNA that was on the gold particle. And there's other ways. But those are maybe the two most common. The second question with the rainbow, it's random. Yeah, well, yeah, I think they stumbled on that technique by accident, but it's random, yeah.
Well, no one's raising their hands.
Me, back here at the bar.
The barman.
Oh.
Heartland, please.
Just, yeah, okay, one Heartland, and anybody wants to be.
Anyone else? Yeah.
No, so back to the calcium, you know, it finds the calcium, it latches onto it, and that forms the GFP. Is that then time sensitive? Does it fade? I saw in the movies it was fading. Is that a time related thing?
Yeah, so there's like about maybe 50 different versions of that protein, and they have different kinetics, which mean they bind calcium slower or faster, and they release it slower or faster. So some of them bind calcium very quickly, but then let go of it very quickly. So if you record, you would see like a bip, but some of them bind it slowly, so we'd see more like a whoop, and they're good for different things. So if you have a very like low to high calcium, the fast one is good, because you have a big change. But if you have a very subtle change in calcium, you want someone that binds it more slowly and kind of integrates the signal. So basically there's loads of different versions of that protein. So now that is called GCAMP. GCAMP is now up to 9, I think. So there's GCAMP 1, 2, 3, 4, 5, 6, 7, 8, 9. GCAMP 6 had 3 different versions, or 5 different versions. So there's loads of these. There's labs that just make those proteins. Yeah. So when you run your experiment, you choose the one you want, and then you try and use it. Yeah. Yep. Oh. Uh-oh. No, thank you.
Let's go with the front first, and then we'll go from the back, and that'll be the last two
Computational
questions.
neuroscientist. Yeah.
People who do this kind of studies, have anybody started correlating that with cognitive behavior to see if?
Probably. What do you mean by cognitive behavior? What like specifically?
Because people, computational scientists, they care about what kind of activity is correlated with what kind of behavior. And traditionally, with traditional recording, that's what people look at. This method is advantageous because you get to look at many of the neurons simultaneously.
So the answer is yes, and definitely. So people do experiments where they will train an animal to do a task, and they will look at the calcium activity in the brain before the task, during the task, and after the task. And the sort of fundamental theory of neuroscience is that when you learn something, something changes in your brain structure. And so probably there's changes in the calcium. So maybe some neurons start firing more, and those are the neurons that are involved in the memory. So this has happened loads. I mean, yeah, is the answer. I'm not so much sure about the fish stuff, but yeah, it is definitely in mice.
Because when you were answering her, you said if you inject them with this kind of proton, the behavior might change, or at least the activity could change. And
yeah, definitely.
How does that affect the behavior?
Yeah, people don't like those questions. They avoid it. Yeah, but that's fundamental question, if you put a protein into the cell that grabs calcium, and the cell is using calcium to do something, do you buffer the calcium signal? Do you change the behavior? Everyone should do that experiment. That's the first experiment you should do with a normal animal and a calcium animal. No one does it. Yeah. Well, people do it, but it's, yeah. Yeah. But I mean, they look normal. They're not just like, I can't do anything. Yeah, yeah, so, yeah.
Let's jump to the back for final question. Very far in the back, please.
I guess I was curious, as same with the last presentation, about what exact questions you have or what answers you're looking for in this experiment.
So if the government is asking medical stuff, Alzheimer's, and we need money. So that's if the government or a funding body is asking. I just quite like the pretty neurons personally, but I think it's fundamentally interesting to actually understand how the brain works, like even how a single neuron works, how it integrates information. You know, just all these questions are fascinating. But yeah, when you have problems in brain function, you have a lot of issues with your synapses, but we don't understand how synapses work. So if we understand better how synapses work, then maybe we can find, you know, drug targets and all this kind of stuff. So somewhere in between those things, yeah, yeah.
Okay, Thomas Chater, folks.
Thank you very much.
Clap, damn it, come on.
Thank you very much.
That's right. One more talk to come. We'll be back in about 15 minutes. It's about 9:40 something. 7-ish. We're going to start at 10 o'clock. Grab your drinks now. The bar will close after the last speaker starts, so go ahead and grab your drinks now. If you have any drinks, empties or anything, please note that the trash cans are in the back corner. What you don't throw away, we'll have to clean up, all of us. Being not you, the actual volunteers. So please throw your stuff away when you're done with it. But yeah, come back at 10 o'clock. We'll see you very shortly. Come see us again December 7th. We'll be here at Nagatacho Grid again. Come hang out. Thank you. 10 o'clock. It's about that time. Y'all about ready? Thank you all for settling down, being calm, being quiet, sheep-like humans. Thank you very much for your cooperation in that regard. We are now ready for our third talk of the evening, after which we will release you to go home to your, I guess, lives is what you have. We have these two. Oh, I have a clicker. This is excellent. Now I have to hold, I just realized. No, it's cool. I'll take care of it. So I have 3 things now. It's not that bad. So, again, please, on your way out, please return your empty trash and bottles to the corner. Be very cool, please, to save us time on the way out. Most of us have last trains. None of us live here, I think. Some of us might live here. I'm actually not sure. Anyway, okay, so we have a couple things we want to talk about besides the next Nerd Night, which, again, if you haven't written this down, my God, what have you been doing this entire evening? So, we've already talked about that. Where the hell were we? Next thing. Yes! Where were we? There we were. So, Nerd Night has taken... Oh. Was that you, Cha-Cha? Wow, speakers jumping ahead of me here. So I was going to say wow, as I was going to say. Nerd Night has taken over. We are in Kansai. We're in awesome places like Osaka. December 1st is the next one. If you're there, check them out, facebook.com/NNKansai. If you're in Okinawa, ever, please stop by Nerd Night Okinawa. It's the same evening as ours, December 7th, so you have a choice. Us or them. Emotional blackmail. We're getting close, but we're not there yet. Sorry if that's sensitive for anybody. My apologies. Anyway, it's facebook.com/NerdNightOki, as in Okinawa. The text was not cut off there, I promise. Their event is on the 7th. So come say hey to them. We're trying to help them out, work with them. If you have any questions about other Nerd Nights around the world, if you're traveling around the world, we can hook you up with them as well. Come see us, come talk to us. We're ready to help. So this evening, we have the help of many excellent people. They're on this thing. In no particular order, Don Warren, human. We don't know where this joke came from. I think it was Andrew. Speaking of which, Andrew Woolner, theater human. Big boss Amanda, put the hands up. Camera human Kai. Vegetable eater in the back, Selena. Yes, I said vegetable. Is it lettuce today still? Sweet potato. It is always something different. I can tell from the photos. Me. Joan in the corner. Who, despite being talented, went to my rival university, Georgia Tech. I am from Georgia. You are terrible. To hell with Tech. And Ian, yo at the bar. And also Don Warren, human, which we got to previously. So where was I? I was going that way. So we are end of slideshow crap. That was the whole thing. I'm leaving them up. Are you ready to put on Cha-Cha slides? Are we relatively? Lion! Where's Lion's photo? I have a really great photo of Lion. Why isn't he up here? Lion, stand up. Where are you at? I can't see you. Did he go home? Oh my God. Lion is this awesome dude. He's great. He helps us out every time. He's so cool. When he comes back in, I'm going to scream at him if the speaker isn't already talking, which is probably going to be happening. So, next talk, final talk of the evening. Thank you for sticking around late on this Friday evening. Our next speaker, please forgive me for the mispronunciation. I will try very hard on this. Ade Irma Suriajaya?
Oh, very close.
Oh, very close. American. Stupid. Also known as Cha-Cha, is originally from Indonesia.
Mm-hmm.
She did her undergrad in aeronautical engineering and applied math in China, and then came here to Japan for her master's and PhD in pure math. Pure, very serious. She is currently in her postdoc program at Riken, and she's into analytic number theory and zeta functions, which she will speak to this evening. She also loves listening to music, as per usual, and she will be giving us some funky math tricks. Cha-Cha, take it away.
Thank you so much for the introduction. And oh, yeah, I need this.
Actually, is that?
And yeah,
that's the separate.
I'm really happy with his pronunciation of my name. But if it's too, if you find it too difficult for you to pronounce it or to remember it, you can just call me Chacha. And you can ask me why it is later, because I don't want to spend like 1 minute explaining. And then yeah, I work in the same research team with Don Warren here. So yeah, that's how I got into Nerd Night today, and I'm very happy to try to teach a little bit of math. And thank you for staying until 10 p.m. to learn some math. Okay, so because it's in the title, so let's start with this question. What is infinity? So like, I hope, everybody heard of this before. Yeah, everybody knows how to write infinity, right? Yeah, it's this one. But what is this? So, well, it's something like this. Please excuse my very terrible drawing, and you will see a lot of this today. So it's just like when you're standing on a road that seems to never end. Like it's endless, unbounded. It keeps going on like forever. So it's something like infinity. And what we're going to see today is, now, if we're adding such numbers, if we keep adding 1, 2, 3, 4, 5, and so on, of course, we should go to infinity. It's never ending. But I'm going to tell you today that it can be finite. It can be -1/12. But how come? So here, my most favorite thing in the world, Zeta function has to help us. And okay, then now you will tell me, okay, Chachan, probably you care about that, you like it, but how do you want us to listen to your talk and listening about Zeta functions? But, well, just now Thomas was counting Nobel Prizes, and I think no number theorists have ever won a Nobel Prize, right, Mehmet? Okay, so there's a Nobel Prize here, but you still have to thank Zeta function. All your information, when they are saved electronically, they are saved because of the Zeta function. So I hope you stay awake and see how this happens. So yeah, we are in this state, I'm really sorry for my terrible show. So you can log into your account, like wherever you are, even though a lot of people talking here and there, singing, and maybe people pass by, your information is still safe. And it's all thanks to the Zeta function in the first place, okay? And now, another reason why, I came up with this idea because I was asked, what qualifies you to give this talk? I didn't start with math, and then, I got into math, okay, because I really like infinity. So I thought, maybe I should be one of the right persons to try to convince you how interesting this is. So this is what I was trying to do when I entered college. So I was trying to make an aeroplane. So I was in this university that was introduced just now. So this is a perfect place when you want to study aeronautical engineering, and there I was. But just after a few weeks of calculus, I just love my calculus class so much, and especially when we started with these integrals. I can just spend the whole day calculating integrals. So I thought, okay, I have to do math. But, okay, I was in the university where they didn't have pure math. I don't know if they have it now. And then luckily, I got a scholarship to come to Japan in my 3rd year for a 1-year exchange program, and this is where I was, in Nagoya. So I thought, oh, this is the only chance for me to escape from engineering and do math. That was terrible. They only did everything. At that time, back then, they only had everything in Japanese. And where are the formulas? Nothing. They explain everything in words. Now, how are you supposed to understand all of this? And I thought, okay, now my dream is gone. I already told myself, I'm going to be a scientist since I was 12 or 13. And I'm happy with my decision. And then I ended up going to an engineering school, which is not right, but in Indonesia, everybody told you, even your school teacher told you, if you want to be a scientist, there's no more science to do if you're not an engineer. If you don't study engineering, well, Chacha, you're so stupid. If you go to engineering college, of course, you become an engineer, but we didn't know that. And finally, I found something that I really wanted to do, math, but no, my dream was crashed. But I thought, okay, so because this is something that I really wanted to do, I should not jump off here. I should try. Okay, so the first semester I tried to at least now first overcome the Japanese and do math. And this time I really loved my set theory class, and there I met infinity. And this was so awesome that after I left Japan that time to go back to China, I was looking only for books about infinity, and I always ended up looking at this equation. Like, oh, this is so awesome. I mean, you keep adding like such beautiful rational numbers, and you get pi. Like, you get a unit circle somewhere. Like, and there are a lot of different proofs to this. But like, this is one of them. I was so happy, like, I'm gonna do this. So here, I am trying to convince you how beautiful infinity is. So now, I mean, we go back to basic arithmetic. If we keep adding numbers, of course we get infinity, right? I keep adding things. So now if I add more, right? I mean, I'm just adding more. I add 2 to 1, and then 3 to that, and then 4, 5 further. So I should still go to infinity, but so it's something like this. I mean, I was trying to draw a better one, but then I got lazy. I mean, suppose you have an apple, and then somebody give you another apple. So you have 2 apples. And then if you get another one, you have 3 apples. And if you continue, you will get infinitely many apples. So this is the principle, and it looks right. But how can this become -1/12? This is very weird. But let's see, maybe we don't really understand infinity. Okay, let's look at this. If we keep adding, we go to infinity. But now if we keep losing, okay, you give away your apples, then you will have no more apples. And then you still want to give away apples, then you have to borrow some apples, right? You will be in debt. And like if you continue that, I mean, endlessly, then you will go to -infinity. I mean, you go to infinity in the other direction. Okay, but now what if I add them? Okay, I keep getting apples, like, forever. But then I don't want them anymore, and I keep giving them away. What should I get? I should get 0. I mean, it's infinity minus infinity. But wait, if we change the way we think about this, what if I change the order? I mean, I get one, I immediately give another one. And then I get another one, give another one. Okay, let's look at this way. Okay, now I get one, I quickly give it away to somebody. And after a while, I get another one, I give them. If you keep doing this, I mean, you have nothing. You keep having nothing, right? So if you keep having nothing, of course, even though you continue this forever, you will have nothing, right? But suppose that I still want 1 apple. I keep 1, okay? Now when I give it to somebody, immediately I take another one. And then I keep continue doing this. So I'm keeping 1, and then like keep losing but getting back again. So then, I mean, this will continue forever, so this should be 0, and I end up with 1. I mean, they should be the same, but they are not. Now if I change, I start with a debt. Okay, I don't have an apple. But now, I mean, I just switch the order here. So I started with -1, and then +1, -1, +1. And then, I can start with a debt and just continue having debts. So whatever I get, I quickly give it away to somebody. Then I end up with a -1. So what is wrong here? I mean, they should be the same, but I mean, it depends on how you look at it. You have different answers. The first lesson here. If you keep adding 0, it's not 0. I mean, you have nothing, and then you keep doing that. Of course, you have nothing, but that's not true. That may not be true. And then, whatever thing we did just now, we changed the order. I mean, we were supposed to add everything first, and then losing things. But it's not the same as adding, subtracting, adding, I mean, getting, losing. And whatever thing we did just now, I mean, they can be equal in some circumstances, but it's not in this situation. So this is not right. I mean, they are not the same thing. That's why we got different answers. And the second lesson that we learned today is be careful of the order. You don't just mix the order when you don't know where you're going to. Okay, so now, there are things beyond this dot, dot, dot. So like infinity is really frustrating. I mean, it's also frustrating, not only for biologists, I mean, and physicists today, they were frustrated with some stuff we were also. So what is there beyond this dot, dot, dot? So now, because I want to try to prove this, but I will give you a hint. If I add less than this, now, I mean, I'm not adding 2 to 1. I'm just adding half. So I have an apple, and then my friend cut his apple into 2. I get that other half, and then I get another 1/3, 1/4. It's much less. But this really goes to infinity, and this is true. I mean, this is proven. This is true. This doesn't make sense, right? So I want you to try to guess like how we got this. I mean, this should be wrong, right? If you look at it this way. So now let's look at an example of a very bad math. This is very easy. Let's go back to elementary school. Now, A is 1, B is 1, okay? So they are same. We're good? Now, I multiply both sides by A. Of course, they are still the same, right? We're good. Now I subtract both sides. I subtract this B times B from both sides. Of course, they are still the same. Okay? Now I take out common factors. I mean, you see this. I mean, there's B here, so it's A minus B times B, right? And just trust me, I mean, you can take a piece of paper there and try to check this is true. So, but what happened here? Oh, I have A minus B on both sides. And that what did your school teacher tell you? Cancel them. Be careful, I really hate this word. It's not cancellation. I mean, we mathematicians say a lot of this cancellation word, but we know what we mean. I mean, like, even though sometimes mathematicians also don't know what we mean. So when you don't know what you are canceling, don't cancel. Okay, now, so what you wanted to do was you divide both sides by this factor, right? Okay, so now we are good, right? We were multiplying both sides by the same thing. We were subtracting the same thing from both sides. Now we divide both sides by this A minus B. What happened here? A plus B is equal to B. A plus B is equal to B, so 2 is equal to 1. That's why I told you don't cancel things when you don't know. So we were good up to here. And the problem here is that now if you plug in A and B here, you know that it's 0. You don't do 0 division. So you don't divide both sides by A minus B. So A plus B is not equal to B, and this is true. So the correct answer to this equation is 0 times 1 plus 1 is 2. So now 0 multiplied by anything is 0. So you get 0 equals 0. We were good. We were just bad with the cancellation. Okay, so now, how do we prove this? Now somebody there will tell me, Chacha, you did a bad math also here. That's why you got a very nonsense answer. Now I will show you another example. I can prove this like more mathematically. Oh, trust me, this is true. Okay? It needs time to prove. This is true. Okay, now I can use it under this condition. Okay? Oh yeah. Now I learned a lesson. X equals 1 gives 0 here. I don't divide by 0. Okay, Chacha, I learned your lesson. So X equals 1 is not allowed. But what's wrong with minus 1? Let's violate this rule now. I mean, let's just forget this condition. If I plug in minus 1 here, I get this number right here. Okay? This looks similar to the thing that we wanted to do, but now we are subtracting something. I mean, we don't keep adding, but like it's 1 minus 2 plus 3 minus 4, so we alternate the signs. And then on the right-hand side, we plug in minus 1, we get 1/4. Now something weird already going on here. Okay, now, this is what we wanted to know. I name it C. I mean, I can't do that, of course. I just name it. And then now, I don't know whether this is a number or not. I mean, you don't multiply a book. I mean, what do you get? Book multiplied by 4? No. You get 4 books if you multiply the number of the books. I mean, I don't know what this is, but I just multiply it. Okay? And then I subtract them. So I get, now, in this order, now again, I violate another. I told you already, be careful with the order. But now I want to subtract it, not like adding this and then minus this, but like this. 1 and then plus 2 minus 4 plus 3 minus 8, minus 0 and then plus 4 minus 8. This is what we get. Oh, I saw this just now. It was 1 over 4. So what happened here is we get minus 3 times c is 1 over 4. Now, oh, this is not 0. I can divide both sides by minus 3. So I get this one. So we got it. 1 plus 2 plus 3 plus 4 plus 5 and so on is minus 1 over 12. But, oh. And, yeah. So now, but these two were infinity. So this should be infinity. But I want to tell you, this is not wrong. I mean, we were doing bad math. It's true. We were doing bad math. It's not the right proof. But this can be correct in some sense. And I will tell you what sense. Now, probably we just didn't understand infinity. I told you already, this is very frustrating. But now, if we use the zeta function, something changes. So Euler, I hope that most of you have heard of him. My favorite guy. Yeah, he passed away a long time ago. And he was learning math from Bernoulli. But I want this Bernoulli. I mean, there are a lot of Bernoullis. I know. I mean, whatever Bernoulli you know. In math, at least, I know these 4 Bernoulli. I mean, they are brothers. They are sons of Johann. And yeah, anyway, I mean, there are a lot of Bernoullis. But Euler was studying math with Johann. And then there's another guy, Pietro Mengoli. I hope I know how to pronounce this correctly. But he was proposing a problem. What is the sum of this guy? Okay, now I have a square here. Like, it's not only 1 plus 1 over 2 plus 1 over 3, but now I square all the reciprocals. What is this? And then Jacob Bernoulli wrote about that somewhere, I don't know, like probably, like I don't know, it's in the history and nobody knew. Like, and then probably he told Johann. I mean, it's written in the book, like probably, like we don't know the truth. And then probably Johann showed it to Euler. Probably, like, we don't know. But what happened is that Euler started thinking. So this was the problem. Oh, I know the answer. This is the answer. I'm like, what? I mean, you are only adding reciprocals of numbers, and how can you get a unit circle here? How can you get this pi? I mean, this pi is, I mean, if you know this is an irrational number, right? This is a rational number. These are all rational numbers. But I mean, the problem is like, how do I get like a very beautiful number here? It's not like, you know, some number over some number, but some weird number. So now if I replace these 2 here with just anything, okay? Now I tell you, as is bigger than 1, it's fine, okay? So now we call it zeta. We call it zeta. We can write it like this. I mean, you just trust me, you can write it like this. It's a reciprocal, so it's a power of a minus, of a negative number. And then now, if s equals 1 is allowed, what do we get here? It's 1 plus 2 plus 3 plus 4 plus 5. Oh, this is the thing that we wanted. But if s equals 1 is allowed, well, there, Riemann comes into action. So that's why it was called the Riemann zeta function. So he made zeta meaningful for any other s, okay? But just don't put in 1, okay? 1 is not fine, but other than 1, it's fine. It's meaningful. Now we have zeta function. So, but the problem is that it's only written like this only when s is bigger than 1. Only at this time. But then we need some tricks to make it meaningful. I mean, it's bigger than this, but like I don't want to talk to you about complex numbers. But anyway, it's at least for s bigger than 1, this is fine. So the point here is that zeta minus 1 is not equal to this one. Okay, we have zeta minus 1, but this is not this one because this is when S is -1, but it's not allowed here. Okay? But zeta -1 is indeed -1/12. So this is the number that we wanted. So this is true. I mean, we can prove it. So now, the answer to this problem is that zeta -1 is -1/12, and if we write it formally in this way, okay, then we get this one. So the answer to this problem is that this sum formally goes to -1/12 if you keep adding them formally. So now, again, to end my talk, you think, oh yeah, this is just so complicated, I don't care about that. I want to remind you, this is important. Your bank account information is very safe. Because any encryption now that works nowadays, so far, they all come from this method. But the problem is, how? I'm not going to teach you anything. I'm not a cryptographer. I don't know anything. But I just know that it helps because Zeta can detect the prime numbers. I mean, prime numbers are numbers like this. And, I mean, detecting these two guys prove that, oh, even though these prime numbers, they are so complicated, it's so difficult to pick a prime number. So it's difficult to find a prime number that we want. But Zeta can approximate the location of prime numbers. Okay. And then with that method, finally we get the current crypto system. So now, electronically, everything can be encrypted safely because we know this fact. But I want to make so now, I mean, we are in this state, like we are in a happy life, we are safe. I mean, so far. I'm not saying like forever, I don't know. But I want to make a last remark here. The crypto system itself was not invented by mathematicians, unfortunately. But they used this idea about zeta function, so somebody proved this, and then they used it, and then they invented this encryption keys for computers and stuff. Now, I was told to give some information about myself, so if you want to know more about me, just Google my name. This spelling should be unique, as far as I know. To my knowledge, it should be unique. So if you find this name, it should be me. I mean, unless somebody else has her daughter named the same. Or you can write an email to me. It's the same name without spaces and capital letters. And if you're still curious, maybe you can find some information on my website, but I forgot to update it so the past month. So something are not there. And thank you so much for your attention.
I'm really looking forward to the questions.
Here's a question.
Do you personally expect ATIA to produce the missing parts of the proof of Irma hypothesis?
Oh, that's a very good question. No, I mean, I don't want to try to do those. I mean, I don't think I can do, and I don't want to be doing false proofs. But like, most of us believe that we need revolution in our math now to do that. So that's why you still haven't found the right proof.
Can I ask a really stupid question?
Mm-hmm.
Is 0 a number? Or is like 1 prime? Or is 0 prime? Or is infinity prime?
What do you mean? Sorry.
Is 1 a prime number?
No.
Is 0 a number?
No. 0 and 1 are numbers, but they are not prime numbers.
Is infinity a prime number?
No. Infinity is not a number. Thanks for your question.
John back there behind them.
Yeah, we're going to do it.
I have another question.
Yes.
So you said that if you add 1 and 2 and 3 and 4 all the way, all the numbers you'll add up to infinity.
Yes, should. It should.
But then I have a separate question. But that means you're adding up to infinity, but then that means infinity is smaller than infinity because...
That's a very good question. Yeah, this is something that I decided to not talk about today because actually infinity, I mean, I just gave a hint just now. Infinity is not a number. And it's not a unique unit, I mean, if you call it. So that's why there are different infinities, and there are infinities which are bigger and less than other infinities. Thanks for your question.
Okay, Lana and then. Okay, so very basic question. Why is it called zeta function? It could be f, it could be a. What's it?
That's a very good question. Actually, we also want to know why somebody called it zeta function. And at some point, and everybody likes it. And like everybody's crazy about zeta function. I'm a zeta function person. Like whatever password you do, you put zeta. Like it's so stupid. But we don't know why.
Yeah, so you showed at the beginning that when you do a summation, infinitely the order matters. And when you're proving when you do 1 plus 2 plus 3 to infinitely, you can get -1/12. But during the demonstration, you had a subtraction, and there was a positioning shift.
That's true.
So can you get other answers to the same summation if you do the positioning differently?
Yes, because it's not the right proof. So actually, we can manipulate that and do stupid things, but because
and
that's
get other
wrong.
numbers.
Yeah, probably yes. I mean, I don't quite remember, but it's possible because it's not right.
So this summation can be equal to different
yeah, it depends on how you look at it. I mean, like so far, we only know it's like this. But it should be true because of the zeta function. I mean, because we get this -1/12 exactly because it comes from the zeta function. But it's like very mysterious. Mehmet, you're not going to ask me questions.
I don't have a question, but I have a joke.
Okay, yes, please do.
So why can't you cancel zeros from both sides of an equation? Like a times 0 equals b times 0. And why can't you cancel zeros? It's a joke.
No, I mean, okay, I do have an answer. Okay, I do have an answer to this, but I don't.
It's a biology answer. It's an evolutionary answer.
Oh, please tell me.
Once upon a time, there were zeros that you could cancel, and there were zeros that you couldn't cancel. The zeros that you could cancel got cancelled away. Now you're left with zeros that can't be cancelled.
That sounds so true. No, it's not true. Thank you for your joke.
Do we have any further questions? Some on the risers.
Thank you. So you showed us a couple of neat results from the zeta function. If you use -1, you get -1/12. If you use 2, you get this weird irrational thing.
Yes.
Are there any other fun gems in the zeta function
A
you'd
lot.
like to talk about?
Yes. At least like for all positive even integers we have, and then we also have a lot of stuff that give you 0.
That's all she wanted to talk about.
What? Do we have one final evening ending question?
No pressure.
Back in.
Yeah, because we have to end the night. We should not go to infinity.
Is this the final question?
It is indeed.
Yes. I have a question for the organizers. Why did you leave the hard one till the end? That's my question, thank you.
I'm sorry, now I feel bad. Like I didn't.
Organizer, you're drunk enough you don't care.
I didn't want to make it hard.
Save the best for last.
Oh.
Thank you, John. All right. So you said, all right, you're not a cryptographer, but I'll ask something about cryptography. From what I understand of cryptography,
yes,
it's all about finding whether the other prime factor of a very, very large number.
Okay, yeah, if you want to break it, you mean? Okay.
Yeah.
Okay.
Right, exactly. And you said that zeta functions can maybe be used to actually find where a prime number might be. So does that mean that a zeta function could actually be used to try and help you break?
I think so, if you try hard enough.
Okay.
Because there is something non-trivial there that people never tell you.
Okay, all right. So a little bit of sweat and lots of hours at the calculator.
Yes, yeah.
Okay.
And good computer.
Yeah, I'm just like, put that in the memory. Okay, I think. What? No, I was just going to say that was the last question.
It was. My God, it was. You're in my way. I got to run right past you. Come on. What's going on? All right, all right. Thank you very much, all, for coming. Thank you very much. Cha-cha. Come back and see us again, December 7th, 2018, seeing as how it's the same year. I don't know why they said that. We'll see you again next month. We're here back at Nagatacho Grid for Nerd Night Tokyo as per usual. If you're in Okinawa, go hang out at Nerd Night Okinawa. But if you're not there, you should be here, damn it. So we'll see you next time. If you want to hang out, some of the speakers are still here, but probably leaving soon. Come talk to us if you want to do a talk with Nerd Night Tokyo in the future. If you want to talk about the grants, feel free. Talk to Amanda, actually. No idea about that. Yes, if you want to post /fic, oh my God, don't tell me about it, please. Anyway, thank you all for coming. We'll see you next time. Good night.